Okay, so now we've done our transcription and our translation, so at this stage we have a nice little chain of amino acids. We need to move on to protein folding because that's essentially what all of this is about. We're trying to get, from our genes to our proteins, we're trying to get something functional. So this is our result from the previous slides, from the previous things we were looking at. But what do one of these actually look like? What does an amino acid look like chemically? And this is important. So essentially you have a central carbon, a hydrogen. You then have a carboxyl group, and this looks like this. So you have your C, an O, and a H, and then you have a double bond and an O down here. And then over here you have a nitrogen and hydrogen. And then here you have the very important R group. So we'll just label these up. So here, this is called our carboxyl group. This is your central carbon. Ooh, not drawing in those. This is your simple hydrogen. This is your amino group. So this is your amino group. And this is your R group down here. Ooh, different colours. Okie dokie. So this is essentially your very basic amino acid. Now, this stays the same for every amino acid. So the carboxyl group, so same, is the carboxyl group. The amino group, the hydrogen, and the central carbon. So what's different is the R group, and this is very important. Now this is what infers the amino acid's characteristics, okay? Which is something we'll go on to. So for example, glycine, I'll draw up glycine so you can see what I mean. So we have central carbon, your H, your carboxyl group, H, double bond O, and you have down here your N, H, H. And your R group down here, and this is the simplest amino acid, okay, is another hydrogen. Okay, and this is glycine. So this is the amino acid glycine. Now, another one, alanine, is also kind of simple. That's a bad bond. C, O, H, O, O. And instead of a H, we have CH3. So just quickly draw that out. And that is alanine. So you see that the only thing that varies between the two, other than my bad drawing, is this, which is your R group. And this infers the amino acids. Ooh, don't know what I'm doing there. Amino acids. Characteristics. Okie dokie. So now we know what an amino acid actually looks like chemically. Put that back. We're going to move on to the primary structure of the protein. So we've kind of got the important bits. We've got our amino acids together, and now it's time to fold up the protein. Okay. So here, this is actually our primary structure. We've already done it. It is a string of amino acids joined together by a peptide bond. So we're already there. We've already got the primary structure. We've got that down to a T. So a peptide bond is a covalent chemical bond formed between two molecules when a carboxyl group of one molecule reacts with the amino group of the other molecule and it releases water. So we can see this here. So we have the carboxyl group, we have the amino group, they're going to join, and there goes your hydrogen. And here we have what's going on up here. We have exactly the same, and then this is the peptide bond, and you've got your amino acids being joined here. So primary structure, we've already got that down to a T. Secondary structure, it's always going to get infinitely more complicated. So the secondary structure of amino acids are determined by their backbone interactions. So these are hydrogen bonds. Okay. So you have one of two. You either have your alpha helix or your beta pleated sheet. Now I am horrendous at drawing, which is why I've put images in there. So I will just zoom in to show you. You can see with the alpha helix, you have your hydrogen bonds joining the alpha helix, and that stables the secondary structure. And with your beta pleated sheet, you have the same thing going on here. So they're also stabilised by hydrogen over here. So your hydrogen bonds go across here, stabilising the structure as it goes. Okay. So that's your secondary structure. So you've already got that down to a T. So primary structure is determined by peptide bonds. Secondary structure is determined by H bonds.